Have you ever wondered how a complex organ actually, knows how to build itself from scratch?
Oh, it's a huge question.
I mean, really think about it. If you were handed a trillion microscopic building blocks and told to assemble a fully functioning heart or a kidney or even just a patch of skin, where would you even begin?
Right, you'd be completely lost.
Exactly. We tend to look at the human body as this finished, polished machine. But the invisible choreography required to actually construct it is just mind-boggling.
It really is. And, for decades, biologists have been desperate to watch that construction process happen at the molecular level. But they've always hit this massive, seemingly impossible wall.
Which is what, exactly?
Well, you typically have to make a really painful choice in biology. You can keep the spatial organization, meaning you know exactly where every cell is located in the tissue, or you destroy that 3D structure by grinding it up.
Right, grinding it up to get the deep, high-resolution molecular details about what the genes inside those cells are actually doing.
Exactly. You just couldn't have both at the same time.
So it was either a highly detailed map of a broken city or like a blurry satellite image of an intact one.
That is a perfect way to describe it.
Yeah.
But today, we are jumping into a deep dive on a groundbreaking 2026 paper from the journal Cell by Asami and colleagues that completely upends that biological trade-off.
Because they found a way to get the spatial map and the molecular data at the exact same time.
They did. And they did it by looking at the hair follicle.
Now, I know hair follicle might sound like we're just talking about your latest haircut, but we have to reframe this immediately for everyone listening. The hair follicle is essentially a highly complex mini-organ.
Absolutely. Is the absolute perfect model system for how all major organs develop.
And that is exactly why they chose it for this mission, right? To explore this new frontier.
Right. This research team figured out how to map organogenesis, which is the formation of an organ in 3D space, and then they mapped it across the fourth dimension of time.
Which is just wild to me.
And the most staggering part, they extracted this entire 4D movie of an organ's lifespan from a single static snapshot of tissue.
Okay, that immediately raises a massive mechanical question for me. How do you get a moving 4D timeline from a static 3D freeze frame? I mean, you can't just invent time from a photograph.
Well, you actually can if you understand the magic of a technique called 3-deep, combined with something they call spatial pseudotiming.
Okay, let's unpack this. What exactly is 3D?
So 3-deep stands for 3D DNase Enhanced Expression Profiling. To understand it, we have to look at the exact mechanism. The researchers started with a piece of neonatal mouse skin.
Okay.
And to study the intact tissue, they first cast it in a specialized hydrogel. This acts like a biological scaffold, locking every single cell perfectly in its original position.
But the tissue is still completely opaque at that point, right? Like it's full of cellular junk blocking the view.
Highly opaque. A cell is incredibly crowded. It's packed with structural proteins, lipids, and just a massive amount of genomic DNA. If you want to see the active genetic instructions, the messenger RNA, you really can't see through all that genomic DNA. So the 3D deep method uses specific detergents to wash away the lipids, and enzymes, specifically DNase, to obliterate the genomic DNA.
Wait, but if they are blasting the tissue with enzymes to destroy DNA, how do they not destroy the RNA they're trying to read?
That's the brilliant part. DNase is an enzyme that specifically targets and cleaves the phosphodester bonds in double-stranded DNA.
Oh, and messenger RNA is single-stranded.
Exactly. So the enzyme acts like a highly trained demolition crew. It walks into the cellular factory, completely dismantles the massive physical archives of the genomic DNA, but leaves the active single-stranded RNA work orders totally untouched on the factory floor.
So if I'm visualizing this, it's like we're taking a foggy, cluttered window that you can barely see through, and by selectively dissolving the heaviest molecules, we turn it into crystal clear glass.
That is exactly it. Because the tissue is now optically and chemically clear, fluorescent chemical reagents can diffuse hundreds of microns deep into the intact tissue. Yeah, so instead of slicing the tissue into fragile 2D ribbons like we used to have to do, they could read the RNA signatures in their original undisturbed 3D environment.
Okay, so that perfectly explains the 3D map. But that still just leaves us with a highly detailed, clear photograph. I'm still caught up on the fourth dimension. How are they extracting time from this?
This is where the computational brilliance of spatial pseudotiming comes in. When they took that 3D snapshot of the mouse skin, they didn't just capture one single hair follicle.
They captured a bunch of them.
Hundreds of them in a single frame. And because biology is messy, those follicles are all growing at slightly different rates. So that one snapshot contains follicles at every conceivable stage of early development.
That's wild. It's like taking a single massive panoramic photograph of a crowded playground.
Right.
You've got, toddlers in the sandbox, 7-year-olds on the swings, teenagers playing basketball. You aren't watching a single person age in real time. But if you pick them all out and line them up by height and maturity, you can create a highly accurate animated flipbook of human aging.
That is a phenomenal analogy. And to actually do that lining up mathematically, they rely on two specific algorithms. The first is called Bazor.
Bazor, okay, what does that do?
Since the physical cell membranes are gone now, Bazor looks at the clustered physical locations of the mRNA molecules and mathematically draws a precise 3D boundary around what used to be a single cell.
So it reconstructs the borders.
Right. And once they have the individual cells isolated digitally, they use an algorithm called Slingshot. Slingshot looks at the transcriptome, the complete set of RNA work orders, and compares them.
And that plots the timeline.
Exactly. It plots a mathematical curve based on transcriptional similarities, essentially assigning a molecular age or a pseudotime value to every single follicle.
Okay, so let me do a quick summary of this first leg of our journey for everyone listening.
Go for it.
Bullet point 1. 3D clears the tissue by removing genomic DNA, letting us see deep RNA like a clear glass window. Bullet point 2. Spatial pseudotiming uses algorithms like Slingshot to turn a static 3D snapshot of hundreds of messy follicles into a moving, mathematically ordered 4D timeline.
You've got it perfectly.
So, listener, here's a question for you to chew on. If you could freeze frame and 4D map any organ in your own body to see exactly how it was built, which one would it be?
Oh, that's a great question.
Because what this timeline revealed about the hair follicle completely rewrites our understanding of how things are built. Traditionally, biology textbooks break hair follicle formation down into 9 visual histological stages, right?
Right, just based on what scientists could see under an old microscope. But human eyes are pretty easily deceived by physical shapes.
Yeah.
When the researchers applied this new 4D molecular map, they realized those 9 visual stages are actually an illusion.
Wait, really?
Yeah. Genetically, the organogenesis boils down to just 3 distinct molecular stages. The cellular programming is actually operating in 3 major acts. Let's look at stage 1, which happens from pseudotime 0 to 0.3 on their scale.
So the very beginning of the developmental curve. What does the tissue actually look like here?
Physically, it looks incredibly simple. The researchers call this the stage of axis patterning and stratification. You basically just have a dermal condensate, which is a specialized clump of cells at the bottom that will eventually direct growth. And you have the outer sheath keratinocytes, which includes the upper hair follicle cells.
Okay, so it's really just a tiny downward growing tube at this point.
Exactly, just a simple tube.
If it's just a simple tube, what is the 4D map showing us that the old microscopes missed?
The hidden molecular storytelling. Even though the complex physical structure hasn't formed yet, the 4D map revealed that the stem cells and niche cells are already undergoing intense spatial stratification.
Meaning they're already moving around.
Yes. The RNA proves these cells are migrating to their highly specific assigned microzones along the vertical axis of the follicle way earlier than anyone thought. And remarkably, they lock into this specific layout and maintain it all the way through to maturity.
So it's not just a disorganized lump of clay waiting to be molded. It's more like a symphony orchestra, finding their specific chairs on a pitch-black stage, organizing by string, woodwind, and brass, long before the lights come on and a single note is played.
Precisely. They're establishing an invisible blueprint, which sets the stage for stage 2, running from pseudotime 0.3 to 0.7.
And what do they call this act?
This is the stage of structural diversification. If stage 1 was finding their seats, stage 2 is the explosion of complex music.
This is where the rapid-fire construction happens. What specific structures are we building here?
The cells that stratified in stage 1 suddenly differentiate into highly specialized inner structures to form the bulb. You see the rapid emergence of the inner root sheath, or IRS.
The IRS. Got it. What does that do?
Think of the IRS as a rigid internal guiding tube that will eventually force the growing hair shaft to grow straight up. You also get the companion layer, or CP, which provides a crucial structural slip layer.
Like lubrication for the hair to slide past.
Exactly. And remember that dermal condensate from stage 1, it physically folds upward, invaginating into the base of the bulb to form the dermal papilla, or DP, which acts as the permanent signaling command center for the entire organ.
Hold on. Here's where it gets really interesting, is I want to challenge something here. If we are looking at a continuous 4D timeline now, what happens to the old textbook theory about the order of operations?
Oh, I know where you're going with this.
Because didn't scientists think the matrix had to fully generate that inner root sheath, that guiding tubeway, before the actual hair shaft could emerge? The logic was always that you can't push your hair through a tube that isn't built yet.
That is exactly what everyone thought, and it's completely false.
Really?
Yeah, because biologists used to rely on 2D histological staining. The dyes they used would highlight the iris very early. So the assumption was that a fully mature IRS was a prerequisite to induce the hair shaft. But this 4D map busted that myth entirely.
So what actually happens?
By tracking the molecular signatures continuously, the researchers discovered that the genetic instructions for the IRS and the hair shaft turn on almost simultaneously. They are emerging in rapid succession, practically co-emerging, rather than one waiting for the other to be finished.
That is fascinating. It really proves how much you miss when you're only looking at a few isolated frames instead of watching the whole movie.
Exactly.
Let's lock that in with a quick recap. Bullet point 1, stage 1 is all about early blueprints. Stem cells are stratifying into their proper zones before the structure is even built.
Right.
And bullet point 2, stage 2 is the rapid diversification, where structures like the IRS, CP, and DP appear almost simultaneously to form the bulb, totally busting the old myths about timing.
Perfect summary.
So to you, the listener, how does knowing that your stem cells get randomly assigned to their specific zones before the structure even exists change how you view the intelligence of your own cells?
It really is a master architectural intelligence.
It is. But if the blueprint and the bulb formation are impressive, I want to move to stage 3, because this next phase completely shattered my understanding of basic biomechanics.
You're talking about stage 3, which runs from pseudo-time 0.7 to 1.0. The researchers call this the phase of accelerated expansion.
Meaning everything just gets bigger.
Right. All the distinct structures that emerged in stage 2 suddenly undergo massive, rapid growth. But the absolute crowning achievement of stage 3 is the formation of the hair canal.
The literal tunnel through the skin the hair will eventually grow through to reach the outside world.
Exactly.
And if I'm just using common sense in high school physics, I'm assuming this is a brute force mechanical process. Like the hair shaft is made of hard keratinized protein. It's growing upward, so it just physically forces its way through the soft tissue above it like a drill bit pushing through wood.
It sounds totally logical, but the 4D map definitively proves that is absolutely not what happens. The canal is not formed by the physical force of the ascending hair shaft.
The hair doesn't push its way through.
Not at all. Canal formation is an incredibly highly orchestrated chemical and structural dance. It is a microscopic choreography involving 2 completely independent cavities and 3 distinct cellular structures. We have to walk through the exact mechanism here because the coordination is just breathtaking.
Okay, do not skip any details here. Walk me through the steps. How exactly does a biological tunnel form without a drill?
Okay, step 1. 2 entirely distinct cavities begin to emerge in the tissue independently of one another. To create the upper cavity near the surface of the skin, a structure called the UHF supra, a layer of cells in the upper part of the follicle, actually begins to break down.
Wait, so the cells are actively dismantling themselves through apoptosis just to clear out an upper empty space?
Exactly. They're sacrificing themselves to clear the path. Meanwhile, step 2 is happening deeper down. The inner root sheath, the IRS, undergoes its own physical transformation. It grows and shapes its upper portion into a conical shell.
A hollow cone.
Right. And this hollow cone forms the lower cavity. So now you have an upper sinkhole forming near the surface and a lower sinkhole forming deeper down.
But they are separated by a solid wall of tissue. How do they connect into a single canal?
That is step 3, and it is the most surprising mechanism of all. It happens right around pseudo-time 0.9. The 4D map revealed a shocking relative movement. The IRS actually descends deeper into the follicle.
It moves down. How does it move down if the whole follicle is supposed to be growing up toward the skin? Does it, shrink?
No, it doesn't shrink. It's actually still actively growing. But the tissue of the lower follicle beneath it is growing at a much faster rate. Because the bottom is expanding so rapidly, it effectively pulls the IRS downward relative to the rest of the structure.
Oh, wow.
And as this hollow IRS shell descends, it acts like a microscopic vacuum pulling the upper cavity open. The spatial data shows that this downward descent expands the upper cavity massively, stretching it from a tiny 10 microns wide to a whopping 90 microns across.
A 900% increase in width just from this relative downward pulling motion. That is incredible engineering.
It really is. And finally, in step 4, as that upper cavity expands downward and the lower cavity is pulled open, the last bit of tissue separating them simply gives way. The two independent cavities merge into one perfect, continuous canal.
Ready and waiting for the hair shaft to seamlessly slide through.
Exactly.
So it's not a drill bit at all. It's exactly like two independent sinkholes forming in the ground, one at the top, one at the bottom, and then the earth between them perfectly gives way to create a pre-planned tunnel.
That's a great way to visualize it.
So quick summary of stage 3. Bullet point 1. The hair doesn't push the canal open with brute force.
Nope.
Bullet point 2. Instead, the UHF supra, the IRS, and the CP coordinate to form two separate cavities that eventually get vacuum pulled into one tunnel.
Spot on.
So, listener, have you ever assumed a physical brute force cause for something happening in your body, like a pain or a physical growth, when it might actually be a highly orchestrated microscopic chemical dance?
It really makes you pause and consider the rest of the body.
It completely reframes how I view my own anatomy. But this brings us to a really critical pivot in our deep dive today. It's fascinating to know how mouse hair grows. But what is the actual practical application of this 4D map? Like, why map a healthy follicle?
Because a map of perfectly healthy organogenesis gives you a baseline. Once you know exactly how the choreography is supposed to look, you could use it to understand why genetic diseases happen. To prove this, the researchers applied their 3D technique to a specific genetic disease model known as the FOXN1 nude mouse.
FOX1. And as the name nude mouse implies, this genetic mutation leaves them completely hairless.
Correct. They have a congenital anomaly that prevents hair formation. Historically, if a biologist looked at a mature nude mouse under a standard microscope, they would just see a bent, broken hair shaft that failed to penetrate the skin.
Right.
Naturally, they assumed, oh, the physical structure just failed at the last minute.
Right. If the end product is broken on the loading dock, you just assume the factory machine broke right at the end of the assembly line. So what did the researchers actually find?
They took 3D snapshots of the mutant nude mice, used the algorithms to generate a 4D mutant timeline, and then laid that mutant map directly over the healthy wild-type map to spot the exact microscopic differences. And what they found was revolutionary. The hair doesn't just suddenly fail at the end. The 4D map proved that the path to hairlessness begins with organ-wide molecular changes happening long before any physical structure looks broken.
What kind of changes are we talking about? If the structure looks fine early on, what's actually breaking?
Well, it comes down to 3 major disruptions. First, they observed severe delayed molecular progression. By tracking the pseudo-time, they saw that the knockout cells were literally lagging behind schedule in expressing their genetic instructions.
Okay, so the timing is off.
Right. Second, there was vastly reduced coordination among the cells. In a healthy model, the different cell layers constantly signal each other so they hit their developmental milestones at the exact same time. In the knockout model, the signaling was degraded. They were completely out of sync.
Like an orchestra where the violins are playing half a beat behind the cellos. The music is still happening, but the harmony is ruined.
Exactly. And that loss of harmony leads to the third major finding: profound developmental instability. Because the timing was off, the cells couldn't maintain their strict spatial organization.
Oh, so they fall apart.
Yeah, the boundaries between the different functional layers get blurry. That beautiful, precise spatial stratification we saw way back in stage 1 started to physically fall apart. And crucially, all of this microscopic instability happens before a single overt structural defect is visible under a normal microscope.
That is profound. Because, if we connect this to human health, it changes our entire diagnostic philosophy. It means that major birth defects or congenital anomalies like a malformed heart valve or underdeveloped lungs might not start as obvious missing pieces of tissue.
No, they start much smaller.
They begin as tiny, invisible timing errors, a microscopic loss of coordination in the 4D dance of organogenesis that silently cascades over time into a massive structural failure.
It really does shift our entire perspective from searching for broken physical structures to searching for disrupted timelines. It suggests that pathology is often a failure of choreography, not just a failure of mechanics.
So to wrap up our findings on the nude mouse, bullet point 1, hairlessness or any defect isn't just a sudden structural failure.
Right.
And bullet point 2, it is preceded by deep developmental delays and a complete loss of cellular coordination across the whole mini organ.
Perfectly said.
Which poses an incredible question for the listener. Does knowing that instability and timing errors precede physical defects make you think differently about how we might detect and treat diseases in the future?
That is the ultimate promise of this technology, and it's why the hair follicle is such a privileged model system. It's uniquely accessible, yet it undergoes all the immense complexities, growth, differentiation, and massive structural remodeling that your major internal organs do.
It's basically a window into everything else.
Exactly. This cell paper definitively proves that deep tissue spatial transcriptomics is the future of understanding biology. We are no longer limited to flat, static images of dead tissue. We actually have the mathematical and chemical tools to decode the hidden, moving dynamics of life.
It is truly a shortcut to seeing the invisible. And I want to leave you, the listener, with a final provocative thought, building on exactly what we just discussed. Buried in the source material, the authors briefly noted that this developmental trajectory math could theoretically be broken down even further to look at highly specific cellular variations.
Right, like mapping the tiny differences between guard hairs, all hairs, or zigzag hairs.
Yes, which implies the algorithms can handle incredibly granular, highly specific structural destinies. So let your imagination run with that for a second. If a single static freeze frame of tissue can be mathematically reverse engineered to reveal the entire 4D lifespan of a complex mini organ.
Where are you going with this?
Well, could this exact same 3D logic be applied to a single tumor biopsy? Could we one day take a single spatial snapshot of a patient's cancer, run it through this spatial pseudo-timing algorithm, and instantly generate a high-definition 4D movie of exactly how that specific tumor formed, where its internal cells are preparing to migrate, and what it is genetically destined to do next?
Wow. That is a phenomenal question because, if a disease is ultimately just a disruption in the choreography, mapping the exact missteps could tell you exactly how to stop the dance before it spreads.
An invisible choreography happening inside you every single second of your life. Thank you so much for joining us on this deep dive. Keep questioning the mechanics of the world around you, and more importantly, the world inside you. We'll catch you on the next one.